Thanks for having me, Shay. Thanks for RegenWA, and thanks to you guys for being on this call. I know for some of you it's pretty early start, or you've probably had things to do before you got on this call. I see some of you sitting with your cup of tea. I haven't got a cup of tea. It's 6 o'clock at night here in Montana. I've been here now for 3 weeks. When COVID started to shut things down, I got myself here as fast as I could, basically. So my horse trailer, my horse, and now my new puppy, who you might hear in the background, we're all here in Montana. Yeah, so thanks to those of you who have purchased my book. I really appreciate it. The book really came about as a mycelial process of people asking me the question of how is it that when I'm in a coaching situation or consulting, how do you read a landscape and how do you start to do that for yourself? How do you look for what we call the key enabling factors, like what is it that's putting a drag on your system, how to enable health? And I actually took it to some publishers, and the publishers said, oh, look, you know, it needs to be more technical. We need to take your story out of it. And I'm like, you know what? There's loads of technical reference books out there in the world, and I have them sitting on my shelves, and they're very important, but I can't say I've ever read any of them, especially not from start to finish. I'll just reference to them. And what's been amazing is I've had like messages from people saying, I've read your book 4 times. I'm like, who reads a book 4 times? And then, you know, it's all dog-eared and full of highlighters. And that's really what the book was about. Like how can we really enable and empower ourselves so that we don't require, you know, these external inputs in terms of knowledge as well to come in and tell us, you know, how to do stuff. So I've put together a presentation, and then I had some questions, and I've replied to those questions through the presentation, and then I got a whole lot of other questions about 10 minutes ago. So what I really want to invite you to do is to use that chat box down the bottom, and I might skip over some slides just so we've got a little bit more time. But I think it's helpful sometimes to see like images of, you know, what people are doing. And so a big part of the book really was thinking about people's stories and how to share people's stories, basically, of what works and what works well. And yeah, so this is the book. If you haven't got a copy, please grab a copy and share it with your networks. I think there's some really brilliant books out there at the moment on Regen Ag. I think this space is just really blowing up, and part of that is, and you guys know this too, I mean, I think I was in Kangaroo Island when doing some workshops on sort of fire recovery and soil health, and you know, you've come out of a massive, well, you're still in drought, then you get fire, then you got COVID, I think there's something else, and it's like, here are all of these externalities that are putting more and more and more pressure on agriculture, and it's happening everywhere. Like everywhere I go, people are saying, we've never seen a season like this. The markets are falling out of the bottom of things. We're seeing systems that, you know, the industrialized system is really coming up to the forefront in terms of we cannot continue agriculture in the industrial model. It just doesn't work, and it's not resilient when we come under stresses like we are right now. And it's exciting to see many of the people mentioned in the book are really looking at how do we shrink the number of steps it is from, you know, producing food to those that are receiving it. And people are asking the questions of where do I get good quality food? So what I want to talk about is what creates healthy soil. I just like this picture, sorry. It's like, I'm talking to Australians. Yeah, let's put a picture of a kangaroo. Sorry. So yeah, what is it that creates healthy soils? And so we're seeing more discussion around what we call the soil health principles. My number one principle actually is how do we optimize plant bricks or plant photosynthesis? Because that's what's driving the whole system. That's what's building soil, that is what's providing nutrient dense forage, that's what's really building resilience into that system. And so I put that as my number one. Now, how do you optimize that would be the magic question, right? So we're going to talk some more about that. So those of you that don't have a refractometer, I'd really advise that you go and get one. They're not very expensive. Just get them online and start getting into the process of measuring your own plants. How well is this plant photosynthesizing? Are my weeds photosynthesizing higher than my crop or my grasses or my orchard or my market garden vegetables. On this webinar right now, we've got people that are doing market gardens, orcharding, sheep, beef, very diverse land use and ecotypes. So I will try and answer like your more specific questions to a specific ecotype, but at the same time, we've got to keep in mind there is a diversity of people in here. But seeing this is a WA event, we will focus more on WA. Ensuring year-round ground cover, and I'm going to talk some more about that because, you know, summer in Western Australia is not easy. What does year-round ground cover look like? How does that apply? Reducing or optimizing disturbance. So we can think about reducing disturbance, but actually disturbance is a natural phenomenon, and even grazing itself is a disturbance. So how do we optimize that in order to keep our soil building processes happening? How do we lift above and below ground biodiversity and biomass? So how do we maximize what's happening above and below ground? Because that's what's really pumping those sugars down, building root systems and building carbon. And then taking action on what we call the enabling factors. So if you figure out what's putting a drag on your system, now we need to start taking action to deal with that. And at the end of the day, these are the outcomes that we're seeing from following these principles. So an increase in biodiversity, an improvement in water quality, seeing our soil health measures increase, seeing a reduction in your greenhouse gas emissions, an improvement in food quality, and an improvement in profitability and wellness. And there's been some interesting studies in Australia looking at well-being. And for me, that was one of the really interesting outcomes from writing the book, was interviewing farmers and ranchers, and without prompting, that was one of the outcomes that people would talk about. And I actually did a workshop. I don't know if this guy's on the call, so I apologize if you are, but I share this story. I think it must have been about 4 years ago. I was doing a workshop in WA, and I asked the group, like, what do you want? Like, in your heart of hearts, what is it that you really want would just bring, you know, joy to your life? What do you want to see for yourself in your life? And this guy said, I want to have more lifestyle. I want to have more time off the property. I want to be able to go and dive. And so a couple of weeks ago, I get this email from him going, I just wanted to tell you, I've just been diving with whale sharks. You know, my properties, I don't feel like I'm having to just, you know, I'm running to stand still, that actually I do get the time to have lifestyle and spend time with the family. And it was just so touching. It was beautiful. But it's one of the things that we really see. If you can start to have a system that works for you, instead of you working for it 24/7, what does that look like? And well-being is such a big part of that. And that's what that's what really drives me, is how do we get well-being in our systems, not only in the soil, but in our own lives. So I talk about in the book the 5 M's. So what is it that's putting a drag on your production business, or what is it that would enable your business to really flourish? And so these 5 M's are your minerals, your microbes, your organic matter, or OM. I know it's cheating, just let me go with that. Your management or your mindset. Which one of those is putting a drag on the system? And if we think about Western Australia, certainly organic matter is going to be a driving factor. What's happening with microbiology, certainly for some of these properties, being able to really optimize management, especially when you have very large properties. And what we find for most of us is mindset has a big part to play. So yeah, what is happening with your mindset? Do you believe that, what do they say? If you believe you can, or you believe you can't, you're right. And it's one of the things that we really go to work with at Integrity Soils, which is my company, really looking at what's happening with mindset, because often that is your biggest drag on performance. So when we think about what's happening in soil, after plant photosynthesis, soils being able to breathe is your #1 factor. How long can you survive without air? And for some of us, maybe we can hold our breath for a couple of minutes. I don't think I could survive even that long. And it's the same for our soil. So if our soils are compacted, if our soils are restrictive, then nothing else works. And, often if you're working with an agronomist, they're not considering this at all. And actually it's your #1 driver for yield and health. So soil compaction, or tight soils, are driven by multiple factors in terms of what's happening with chemistry, biology, what sort of physical constraints do you have on those soils? So is it your physical elements, your minerals, your microbes, or is it the management that underpins all that the management that surrounds all of that. All right, and if you don't get your management right, you can push along a regenerative ag program if you want, but it's going to cost you a lot of money. So the first place to start really is your management. And so that's going to depend on what kind of sector that you're in, what kind of production have you got. But getting that management right is absolutely key. And then enabling those soil health practices to fit into that. And so looking at soil compaction differently, like actually, is it a major microbial imbalance? So what is it of those 5 M's that's limiting airflow on your farm? So go out, dig holes, see, have you got a compaction layer? That shallow compaction layer, we call the cow pan. You know, is it livestock? So it might be sheep pans for some of you, or is it deeper down like old plow pans? Is actually that with a drag on that. So there's different factors. So your chemistry might be playing a role. So if you've got high potassium, high magnesium, sodium. So potassium and magnesium pull the soil tight. Sodium disperses it, and in the same way, from over dispersing, we create compaction. Or if you've got low calcium, right? And for some of you, that's often what's going on. Some of you have got very, very high aluminium. So having a look at what's happening with aluminium. But that ties into your calcium, right? Is it a physical constraint? So very clay soils, but even clay soils, we can still get them to open up, right? So it's about these mineral balances, biology. Or are you just sitting on subsoil? You know, literally you're just on top of rocks. Okay, that might be limiting air movement. These bacterially dominated soils, or what I call the sleepy soils, where you might have fungi in that system. And one way to tell might be if you are in areas that have a lot of woody encroachment, woody type species, your mulga, your acacia, these are the sleepy soils, right? So they've got more fungi in them than the bacterial dominated soils, but they're not functional, they're not awake. They're needing some kind of stimulant to wake them up, which in the past might have been fire. It might have been like large, do we call it a herd of kangaroos? A herd? I'm not sure. Anyway, they might have had animals moving across those systems and waking those soils up. Or is it low organic matter? In the past, has there been over and under grazing? Because I'm sure that's not happening now. But is your grazing management the cause for why soils are becoming compacted, right? Allowing animals to meander and stay in pastures for a long period of time is going to start to compact those soils. Are you cultivating? We had questions about flood irrigation. So are you creating waterlogged conditions that will then start to compact the soil? Heavy machinery. Now this is interesting, is that just because you have heavy machinery, you know, moving over soils does not mean the soil necessarily is going to compact. And we've been in situations where we've balanced the minerals, stimulated microbiology, and we can have heavy equipment running over that and it won't compact, right? It's not necessarily just about the heavy machinery, but what's happening with these other aspects. If you are using asides, so chemical controls, pesticides, fungicides, herbicides, they can all tend to create compaction because they drive soils bacterially dominated, and I'll talk about why that's important. And then a lot of soluble fertilizers. So think about World War II when they're using anhydrous nitrogen to create landing strips for aircraft, right? So soluble fertilizers, some of the things that burn up your carbon, and then that makes soils tight.
Facebook them.
So I just want to show an example from Western Australia. So I try and make sure that I'm providing new images and new information. So some of you might have seen this before. So for this property, the owner was really excited because he drove up his driveway and he just had like half an inch of rain, and he was like, yes, we've been in this drought. This will be awesome. We're going to get all this moisture. And as he came up the driveway, all the water was coming across the surface and down the hill and causing these erosion rills. And he had that aha moment of, wow, my soil is not absorbing water. So he used the yeoman's plow, which many of you might be familiar with. So the yeoman's just to open up below that compaction layer, and down that he dripped sugar and humic acid. So anytime you're ripping, I want you to drip. Anytime you're doing anything mechanical, think, how do I feed my underground livestock? You're out there anyway. And something like sugar and humic acid, that's only going to cost you maybe like $1 a hectare. Like, we're not spending a lot of money. He also seeded perennial species. So there's some panic in there. There's some ceradilla. There is chicory, plantain, native grasses, and native forbs, right? And with that, he put a seed inoculant, which had Pseudomonas fluorescens in it. I really like Pseudomonas fluorescens. It's one that, it's a type of bacteria that works in combination in the soil. It will actually help to stimulate your mycorrhizae fungi, which he also added. But Pseudomonas fluorescens eats the bacteria that causes ice nucleation or frost. So it's a bacteria that eats the frost-making bacteria. So down to a -6 degree frost event, these organisms will control frost damage in your plants. And we've seen some pretty major frost events in Western Australia. So this would be one of my go-to organisms, but you don't have to buy it as a packaged product, and I'll talk about that in a minute. He also added Bacillus subtilis, right? So biology down with the seed. So this is a line. So on the right is where that treatment didn't happen, and on the left is where the treatment did happen, all right? And unfortunately, when this photo was taken, it was just after it grazed. So you can see what it looked like before it was grazed. This is what it looks like after it grazed. Now, we'd been driving. This was March 2015, 2016. And it was dry, and we'd had 3 days over 40 degrees, so it was dry and hot. And this was really the first ground cover that we'd seen apart from the eucalypts, right? And just seeing a lot of open ground, a lot of real drought stress. So this is an exclusion zone showing, so this is after your 3 days of 40 degrees, you can see how stressed these plants are. But look at chicory. Like, I love chicory, and there's some people on this call from like Canada and New Zealand and the US. Get yourself some chicory, guys. It's, I just love it. We grow it in the hot, we grow it in the cold. I call it my Dr. Zeus plant. You know, I could eat it in the rain and on a train, on a plane, whatever. We just find that it does really well in a very broad range of climatic environments, and it's such a good animal fodder, and it's such a good, it's got a big penetrating root that can really get down through hard pans, and high minerals. Yeah, anyway, it's good stuff. So this video, what I'm showing, and unfortunately I started recording after I'd already poured some water on the left. So the left-hand side, just skip forward a bit. The left-hand side is what's had the treatments. It's got a living growing root in it, and the right-hand side hasn't had any treatments and hasn't really got living growing roots. And so when you pour water on this, the water literally just runs over that surface, and it doesn't sink in at all. And so if we pour water on it and break it open, the water, terrible video, the water doesn't infiltrate at all, whereas the soil on the left, we've got aggregate structure, we've got water absorption, we've overcome those water repellencies, and really that's all happened in 4 months, 5 months. So we did this in October, and we went there in March, October, November, December. All right, 6 months. It cost them some money though, all right? So I'm not saying that this didn't cost money to do, but we can change soils far quicker than what we think is possible. I've got a project in New Zealand, and it's in the book, and we were building 1 millimeter of soil every month. You know, we're doing stuff that, you know, the scientists will come out and say that's not possible. And I'm like, okay, well, that's all very well, but we are doing this in the field. You know, how quickly can you get a rooting system like that? We've got living, growing roots in these, yeah, pretty harsh environments. So pretty exciting, right? Very good. So one of the things that we see in WA is these hydrophobic conditions. So there's 2 things that can create that. It can be a byproduct of organic materials, so the volatile organic compounds. So these are things that are going to come off your gum trees or organic material. You might see this after fire. These organic materials that aggregate around your soil particles and repel water. I mean, hydrophobic literally means afraid of water. If you have soils that are afraid of water, you're in some trouble. They can also move down through that soil profile and then form a barrier to water movement. And I saw this in Kangaroo Island, just soils that there's no water going into that. All that water is just hitting that surface. It might absorb if you've got a little bit of litter maybe on top, but basically any small rainfall event and that's evaporating. So your actual rainfall efficacy in many areas in WA is very, very low. So not only in your low rainfall and heat, but water's not going in. Microbiology also create these hydrophobic conditions. And for those of you that have been watching what's happening with the pasture dieback, and Shay, is pasture dieback in Western Australia? Has it arrived?
Sorry?
Is pasture dieback in WA?
No, not to my knowledge. I know that it's been seen in some orchards though.
All right, so mealybug has been, the fingers are pointing at mealybug. Now, mealybug isn't what's causing pasture dieback. Mealybug is secondary to something else. Like, mealybug doesn't just happen because. But one of the things that mealybug does is it makes these waxy coatings that create water repellent soils. So bacteria, when they get very, very dry, they make this wax to protect themselves from drying out. They make these water repellent conditions. And so you want to get really curious, is your soil afraid of water? And if it is, start to take some different actions. So ways that we can manage these hydrophobic soils is we can actually apply low rates of alkaline products as sprays. So you could do what I call a liquid lime or a fine lime. We make that using the whitewash recipe. It's just hydrated lime or burnt lime, slate lime. We can also use milk. And with the dairy situation in Australia at the moment, it'd be nice if you brought some milk off those poor farmers and then applied it to land. All right, so that will actually help to disrupt some of that hydrophobic condition. We're also using vermicast, so that's a worm poop, basically. And we're drilling multi-species of cover crops with seed dressings of those types of products on the seed to allow that plant to start to germinate, take that materials with it, and break up these hydrophobic conditions. So vermicast, some of you may know that's how I started in my career. I'm highly biased. I love worm castings. You know, worms have remained unchanged for like hundreds of millions of years because what they do is perfect. And what comes out of their butt is the elixir of life. It contains hormones and vitamins and enzymes, and then these microbes. And these microbes specifically which eat hydrophobic waxes. So the Pseudomonas, the Serratia, and Bacillus species, you can buy them as packaged products to break up hydrophobic waxes, or you can just get yourself some vermicast or worm extracts, and I'm going to talk about them a bit. All right, so that's something that you can do. So I want you to consider that the soil environment is what we call a holobiont, right? Which is that every aspect of it is connected, that nothing exists in isolation, just the same as it is with our own bodies. And the discoveries that they're making about the human gut microbiome is that everything comes back to microbiology. And actually, they're now saying that 100% of immunological disorders in the human body come from the gut, 100%. So that's anything to do with inflammation, autoimmune, allergies, so, you know, arthritis, Alzheimer's, all of it's coming back to microbiology. And so this is the new wave of science, and it's really exciting to be involved in Regen Ag and finally start to see this huge amount of research that's coming through to say, yes, everything is connected, the hippies were right. And so it's that the host or the plant and the microbiology together make this holobiont, right? So you or me, plants, soils, microbes, forests, whatever, it's all interconnected. So part of this interconnectedness are these communication molecules. So many of you would have heard the term quorum sensing by now. It was discovered in the 1960s looking at bioluminescence. And they discovered in the early 2000s that it's quorum sensing that's switching on or off disease organisms, let's say even in the human body. So the medical world's been very interested in it, but it's basically just signaling molecules. So that plant is signaling to its microbiology and the microbiology is signaling to it. Okay, so this is constant chatter that's happening in soil. And what we're finding is that chatter or those chemical signals, you only need parts per trillion. So we need very small amounts of this to actually switch a soil on. And it's great to see this research because it's explaining some of the phenomenon that we're seeing on rangeland soils where we're like, we've just put out 2 kilos of vermicast as a slurry and we're seeing plants germinate, or we're seeing dung beetles arrive, or we're seeing life come back to landscapes. And it was hard to understand until we understood a little bit more about quorum sensing. So part of these signaling molecules and how plants are communicating is they have exudates, they have aromatics, there's hormones, enzymes, vitamins, sugars, amino acids, proteins. And every single cell, so imagine like the outside of a bacteria, has hundreds of thousands of receptors. And these receptors are waiting for signals to turn on or turn off. And it will actually change how genes are expressed. It'll change how an organism is able to deal with resilience, or switch on, switch off, provide the plant nutrients, provide the plant with a signal so that the plant can defend itself, right? So it's incredibly complex. We understand just a little drop of what's going on, and it's also very, very exciting. So pretty much if you think about the human body and 100% of that being like immunological disorders relating to microbiome, it's the same in the plant. So plant health and nutrition is driven by biological functions, right? So the more diversity we get in there, the more signals, the more that increased resilience to stress, the better the crop health and quality. And at the end of the day, the higher your profitability, all right? We're having a system that can recover after disturbance or resist a disturbance longer. So anything that we can do to optimize diversity and biological biomass is absolutely critical. So everything needs to be, how do I lift my biological component in the soil? How do I build soil health? Because everything comes out of that in terms of whatever your goals are. So we're talking about bacteria and fungi. So bacteria and archaea. So archaea are not even closely related to bacteria. They were sort of lumped in together, very small, single-celled organisms. Archaea means the ancient ones, so we find them in very extreme environments, and we also find them just in your backyard. So bacteria are important because they suppress disease, they hold on to nutrients, and they form the soil microaggregates, and that's an important thing to think about. They're very important for your nitrogen cycle, they're breaking stuff down, and they're eating more simple sugars, right? So keep that in mind. They eat the simple stuff, so you think about when you make compost, the heat that's generated is being generated by bacteria, right? Eating green leafy stuff or manure or whatever, right? That heat is all bacterial reproduction, right? And breathing. So typically they are eating simple stuff. So the green materials, so that could be your green grass, it might be molasses or sugar or milk, seaweed, manure, urine, fulvic acid, fish emulsion. So there was a question someone asked about fish emulsion and fish hydrolysate, and I'll get to that, all right? So more simple foods are gonna stimulate more bacterial function, okay? Many of your nitrogen fertilizers and many of the pesticides and fungicides feed bacteria, okay? So they're getting that big bust and boom, and we see that often in Western Australia because we'll see what we call the nitrate weeds. So what happens is bacteria make an alkaline exudate, and in that process, the reason that they do that is it enables them to make an enzyme to pull the hydrogen off your ammonia and replace that with an oxygen, which creates nitrates. Okay, and what happens is when you have, so thinking of coming out of summer, you've got dry, bare soil. This process is going on all the time, but the minute that you get a bit of moisture, it's a signal for the nitrate weeds. So pigweed, fat hen, nettles, thistles, marshmallow, thorn apples, Patterson's curse, foxtail barley, your roly-polies. Trying to think what kind of weeds you guys have. Anyway, I'm sure you've all got some of these, all right? Oh, capeweed. You're all like, no, we don't have any capeweed. All right, capeweed. So what happens is that bacteria are building that up, and then there's a bust and boom process and they're releasing nitrates. That's a signal for your nitrate weeds to germinate. So if you're interested in learning more about the weeds and what causes or what is the germination signals, then get a hold of my book. There's a whole chapter in there that really digs into what are the signals for what kind of organism. So bacteria are essential. However, if we have bacterial dominant soils like these ones, this is a photograph of a property in New South Wales that had been growing rice 4 years ago, and now nothing would grow. So bacterial dominance can lead to compaction or that surface crust. And when you get a surface crust, there's certain types of weeds that are going to be able to germinate because the soil's not breathing, or you might see like club moss, lichen, like really primitive stuff. They're telling you that soil's not breathing. Okay, so we end up with high bacterial soils, low predators, and by predators that I mean soil predators like your protozoa and your nematodes, when they are low, we see that build up in nitrates, and we see nitrates in the weeds, and then that can have implications for animal health, human health, and obviously soil health. So if you have a refractometer, get into the habit of actually getting out and testing your plants. And if you see that your bricks is a sharp line and it's 3 or below, it's telling you there's nitrates in those plants. Do not cut that for hay, do not graze it, right? So that refractometer can be a really helpful grazing tool. But also if you were going to harvest those, say like you're harvesting lettuces or something like that for human consumption, you will taste it because it's very bitter. But the refractometer is going to tell you those are nitrates, they're carcinogenic, that's not fit for humans, and it's what puts people off eating kale. Because when you grow kale with full biological diversity, very high bricks, it's super, super sweet and delicious, and you not have the nitrates in it. But as the nitrates come in, that stuff just tastes horrible. And if you measure a lot of the stuff at the supermarket, you'll find a lot of nitrates, right? Very low bricks, very sharp lines. And that's why, yeah, you want to grow your own greens, basically, is the take home from that. So fungi, on the other side of the spectrum, are very important again in disease suppression, retaining nutrients and breaking stuff down. But these guys make the large macro aggregates. So when we talk about soils as having that chocolate cake structure, it's the crumb structure is being driven by fungi. And this picture on the left is actually a picture of my vermicast. So that's what my vermicast would look like. Very fungal dominant, loads, you can actually see the fungi in that system, and the worms just love it. It was amazing. I miss my worm farms. Anyway, those macro aggregates, and you think about those fungal hyphae, they can travel for like, well, I guess in soil, we're talking about hundreds of kilometers of hyphae in a square foot of soil, right? But they're what holding soil together. It's not plant roots, it's actually fungi. So Kim Deans, who's one of the awesome team members at Integrity Soils, who's in Inverell in New South Wales, you guys would have seen the big dust storms this year and last year. Those dust storms are not a natural phenomenon, right? They're coming off land that doesn't have active fungi in it. And what Kim Deans was seeing is fence line photographs where her place is not moving in the wind and didn't move at all, and the neighbors is blowing away, right? It's a biological system that's holding those soils together, and disrupting that system means we're now losing our topsoil, right? But fungi play a big role in yield. So fungi have the ability to access and mine nutrients off rocks. So geomycology, they're actually getting into that rock and helping to release bound things like phosphorus. And what we find is with programs is you start to lift your fungi, we see phosphate levels coming up in the plant, even though the phosphate levels might still be really low in the soil. Don't care, plants got it, right? What they also do is they make rocks, which I think is so cool. So fungi make actual crystals and rocks. So mullowite actually comes from Western Australia. It's a type of rock. It's copper based that's made by fungi. What's interesting about this process is they're drawing carbon down to make long chain crystals. So you've got calcium, say wetterite, calcium carbonate type structures. That's a very long-term carbon drawdown that's happening with fungi. So if you're measuring carbon, you want to be measuring your inorganic carbon as well, because that can actually be changing because you're lifting fungal activity, right? So it's a carbon sink. We find that as fungi increase, your soil carbon increases and respiration decreases. So we get less, the soils are breathing out. So every day soils breathe in and breathe out and breathe in and breathe out. That respiration declines as you get more fungi, and so that carbon becomes drawn down into the soil. And soil carbon is absolutely critical in Western Australia. You need to be thinking, how do I get more carbon in the soil? Because carbon is your bank account, it's your storehouse, it's what going to be holding on to water, right? It is what underlines your profitability. So the David Johnson and New Mexico State University are doing this work, or he was, but at the university still is, showing that fungi to bacteria ratios are more closely related to production and yield than NPK. So side by side, working with chemical fertilizers to fungi to bacteria ratios, as you increase the amount of active fungi, yield goes up. Whereas if you have very low fungi to bacteria ratios, we see those low quality weed species. So your melons, your button grass, your caltrop. Love that one. I made the mistake of saying to some Western Australians I didn't know what that was, and then everybody brought me some. I now know what it is. I don't need to see it again in my lifetime. Lovely. So David Johnson's showing this. As you increase that fungi to bacteria ratio, so 0.04, very bacterial dominated. I've got this screen that keeps popping up, then I can't see my screen. And then at 1.6, you know, you've got equal biomass of fungi to bacteria, and then 3.68, you've got nearly 4 times as much fungi as bacteria, seeing biomass rapidly increase, right? So as we start to drive that towards more fungi. And what's really cool, and this is important, is that fungi make water, literally. So when they're breaking down organic materials, 20% of what fungi produce is water. So if you think organic materials contain carbon, hydrogen, and oxygen, carbohydrates. So that water is released, and the carbon turns into chitin, which is what the fungi make for their own cell walls. All right, so they take those materials, break them down, and what is released is water. And you'll notice this around compost piles, is that there will be like this ring of moisture around the bottom, right? It's actually coming from the fungi, and as we start to increase fungal diversity, we increase that water. How cool is that? All right. So what they need to eat is more complex carbons, the brown materials. So brown grass, cellulose, lignin, chitin. So I've actually got ranches farms that we've worked with who are using things like brine shrimp, crab shell, shrimp shell, oyster shell that they're grinding up and putting that onto soil to stimulate the fungi. Your straw should be, and stubble should be eaten up by fungi, but what I'm seeing in Western Australia is a lot of oxidized, very low nutrient material that just won't break down, right? And it's not breaking down because there's very low nutrition in that. So fish hydrolysates are more complex, right? So a fish emulsion doesn't have the same amount of oils in it, doesn't have the same amount of complexity in it, often be a cheaper product. The fish hydrolysate I prefer from like deep sea fish that have lots of oils. Now it's a waste product, so I can sleep at night. Yeah, when the fishery beds run out, we'll have to find something else, obviously, but let's look after our fisheries. So humates or biochar. So humates are your soft brown coal, prehistoric, 65 million year old carbon sources, so long chain carbon, whereas biochar are made in a pyrolysis machine. Depends on what kind of waste materials they might be pyrolysizing, but it's something we're seeing more and more discussion about, and it's happening in Western Australia. And wood chips. So I'm a big fan of wood chips. I will chip wood chip. I was about to break into song, but I won't. To actually feed fungi in our soils, right? So yeah, there's how that fungi's all holding that soil together. So what you find is in our environments that disease organisms are always present, but what suppresses that disease is having this biomass and biodiversity of organisms out there. So it's absolutely critical. So if you're seeing diseases, then get interested about what's happening with my beneficial army that are out there protecting and defending and looking after those leaf surfaces and root surfaces, right? And it's the statistics on how much mycotoxins and ergot and all sorts of stuff like that that's in our animal feeds, and probably even in our human feed. I don't know that, they're probably putting it to animal feed, is scary, right? And it's increasing because we're not looking after the beneficial microbes, and so now we're entering these mycotoxins into the food chain. So what I wanted to just sideline and talk about just for a second is epigenetics. So epigenetics, or epi, is the Greek word for above, at, or as addition to, and gene means the genetic code, sorry. So epi means it's above that genetic code. So your gene is, the genes give you how you look, whereas epigenetics, your DNA or your genes sequence stay the same, but it's altered by some kind of triggers. Are they environmentals? Is it some kind of signals? But basically, one way to think about it is you are what your mother eats. What was happening to your mother and father, what your mum was eating when you were actually in the womb, can alter how your life goes, basically. So there's a lot of focus on DNA, all right? And we're seeing more and more DNA profiling, and I did it. It was kind of cool. I found out I had Romanian blood, which explains why I do so much traveling. But they're doing stuff like they'll tell you, you have the DNA, like you have the gene for this cancer, or you have the gene for this kind of arthritis, or you know, whatever. So people go, oh, I have the gene for this kind of breast cancer, so I'm going to take my breasts off. Or we're going to then alter your genetic code. And this is big business, people, and we're going to see this coming out in a big, big way. Is this big focus on DNA. And actually DNA, all that DNA is the blueprint, but it's the gene expression which turns those genes on and off. So the DNA for a muscle cell or a liver cell is exactly the same. Okay, it's the same DNA sequence, but there is a trigger that turns on, let's say the muscle gene gets triggered and the liver gene gets turned off, or in the other case, the liver gene gets turned on, the muscle gene gets turned off. It's all signaling, and it's driven by proteins, all right? So the protein or the environment that you're on switches on and switches off. So when they looked at DNA, they were mainly focusing on, you know, like, oh, cool, let's look at what the DNA is and how can we market that. But what wraps around the DNA is a protein sheath, and that protein sheath retracts and closes depending on the signal. So it turns a gene on or it turns a gene off. So yes, you have a blueprint, but then you have the people that are doing the building. And I think about it like, I guess you got a building crew, you know, and they're given this blueprint, but maybe they've been drinking all night or whatever, and then they did something different to the blueprint and the house falls down. All right. It's that signal that determines how something's going to go. So they did this interesting experiment. Why do they do things, terrible things to mice? Anyway, they exposed male mice to a cherry blossom smell, and then they electric shocked them every time they smelt the smell, to the point that, like, you know, they're just these shivering wrecks. And every time the smell comes on, electrocution. They then did 4 generations of their mice. So they did IVF treatment. And then what happened to their baby mice and their great-great-grandchildren, you expose them to the cherry blossom smell and they will start to shake in fear, even though they didn't have any exposure to being electrocuted, all right? That trauma and that fear was inherited. This is epigenetics, all right? It hasn't changed the gene. They're still mice. They still look like a normal mouse, except now they respond to the smell in a fear process. They did a study looking at exposing female mice that they mated them with runts. So it was a smaller mouse, and she was like, oh, you're not that attractive. But anyway, she didn't have any choice. And what they found was she had runt babies, which you would expect because she mated with a runt. But what they found was it had nothing to do with the fact that she'd mated with a runt. She restricted nutrient flow to the baby. Subliminally, I'm sure she wasn't going on purpose, so I'm not going to give you anything. But in the back of her mind, she switched on an epigenetic signal to restrict nutrient flow to that baby. Then what they did is they did IVF instead. So she didn't know what the male looked like, and she had totally normal babies, right? And I see that sometimes with couples and go, I wonder if she restricted. No, I'm not saying that. So in humans, what we see is in the womb, stress and nutrition and toxins can alter and influence gene expression. So every generation of males that have been born since 1975 have half the sperm counts that their fathers did. Every single generation that's being born right now, right? That epigenetics is being affected by fungicides, by BPA and plastics. There's stuff that's going on now in the environment in the last 100-odd years that's changing how genes are expressed. And this is what's quite interesting, is I find a lot of people that I work with have come from maybe working in the chemical industry where they're chemical sprayers, and they're fine, but their kids are not, or their grandkids are not, right? And seeing, you know, it might be autism or autoimmune disorders or cancers, like early childhood cancers, and going, I wonder if that had something to do with what I was doing. And science is really bad at capturing this when it's something that happens over generations of people. But we can see this happening over generations of rats and mice, right? But fungicides have certainly been culprits in what's happening. So many of our chronic diseases, so like rheumatoid arthritis, it's actually your gene expression stops being able to regulate itself. So it deregulates and we end up with chronic diseases. One of the interesting things is if you suffer from early childhood abuse, there's an increased rate of, say, alcoholism or drugs or suicide or poor relationships, not because of that's what you knew as a child, but because your epigenetic profile changes and you're no longer able to deal with stress in the same way. So it's not all bad. So we can alter epigenetic expression through love. I know you're going, why am I here? We're talking about soil, but I tell you this has something to do with soil. But loving physical touch can actually alter how genes are expressed in early life. So, you know, loving children, giving them giving them a lot of hugs, giving them positive reinforcement has a direct link to how they deal with stress and anxiety in adulthood. There's a link between health and well-being. So yeah, well-being, self-care, mindfulness, and meditation are all proven to alter epigenetic expression. So it's not about going and buying or dealing with medical doctors to try and change your genes. It's about how do you switch a gene on or how do you switch a gene off? And there's things that we can do in our day-to-day life that enable us to do that. So what's it got to do with plants and soils? So there are inherited traits for drought resistance, seed dormancy, biological relationships, and how plants respond to heavy metals that are an epigenetic expression, right? The plant's gene is the same, but how it switches on or switches off is driven by this inheritance. Your microbiology, so mycorrhizae, fungi, and bacteria can alter how that plant's gene expresses itself, what its rooting system looks like. Can it deal with heat or cold? How a plant is grown and where a seed is sourced can set you up for resilience and nutrition or not. And it brings together a really big question about where are we buying our seeds, who's controlling the seeds, and those seeds are often grown in high nutrient systems with a lot of pesticides and fungicides that impact the resilience of basically those plants. So the seed microbiome comes, and I'm going to stop for questions. I'm just thinking I'm getting sidetracked. But the seed microbiome, there's actually microbiology inside seeds, and this has been ignored for a very long time. So it's either coming from the mother plant or coming from soil and air. So you see this, like there's microbiology everywhere that's getting into the actual seed so that when you're planting that seed, it's set itself up to survive in this particular environment. So consider the seeds that I'm buying, where did they come from? What was that kind of environment? Did it have a lot of fungicides? Were they suppressing that microbiome or really lifting that microbiome? So the microbiome primes and speeds up germination. So how well those plants are germinating. If they're germinating in, like when you guys are dry seeding, have you got an environment that sets that plant up for success? Having a healthy seed microbiome induces plant defense so that plant's able to defend itself, and especially if you have a rhizosheath like that, I call these the Rastafarian roots, man, right? So we've got these great big dreadlocks. That's really protecting that plant from the moment that it germinates and enables it to defend itself. It's helping that plant to solubilize nutrients, it's fixing nitrogen, it's producing plant hormones. And if you're getting seeds that are from systems that are very low and beneficial microbiology, they can contain plant diseases. So this rhizosheath is absolutely fascinating. And in Western Australia, you need to be really focused on this because it's providing plant protection, nutrient uptake, it's feeding your microbes, building carbon, building resilience. And also what's super interesting, it's buffering against things like temperature extremes. So instead of having these massive fluctuations because the clouds come over or it's nighttime, and you know, you're going from 40 degrees where soils might actually be like 70 degrees Celsius to having a soil that's actually buffered. So it very slowly takes on soil heat or cools off, right? So that's really helping reduce stress for that plant. But it's also having to buffer against pH. So that rhizosheath, if you look at that root system in there, its pH can be 2 units difference from the outside soil. So you might be like, oh, I've got these terrible acid soils and they're like 4.5, but the plant's actually experiencing 6.5 because of that rhizosheath development. So the game in regenerative agriculture really is how do I develop rhizosheaths like that and defend, help my plant defend itself. It'll also stop that uptake of aluminium, see I'm fluent, 2 languages. Aluminium is that if you have bare, naked roots, there's nothing to stop aluminium coming into the plant, and then it's going to be coming into either our grain or it's going to be in your livestock, and now we've got animal health problems, right? So part of what's happening is that plant is expanding is this process of rhizophagy. So we're seeing more and more cool information coming out about this, but 30 to 100% of seedling nutrients comes from the absorption of bacteria at that root tip. So you see that all that blue fuzz. So this here is a root tip of Phragmites. Those little blue dots everywhere, that's bacteria. So as that root tip and the root hairs are actually initiated basically like an irritation from the bacteria, and they expand, they feed that bacteria, they then absorb it, take its cell wall off, which is full of nutrients, absorb that bacteria, and then continue that cycle, right? So they're feeding bacteria, absorbing bacteria, taking the nutrients, and on and on and on it goes, right? Incredibly important. So if we have microbiology in that root or in that seed, we're setting them up for success. So we want to see that we have hairy, dirty roots, which I know Australians always chuckle at anyway, initiated by having bacteria inside the actual cells. So this gives us disease protection, nutrient and water uptake. So if you look at that plant on the top left, a little sunflower, see that massive amount of root hairs in that system? Well, that's going to be giving you all that water uptake compared to the ones that have been hybridized and messed with on the right-hand side. So if you look at the bottom, ones on the left, they've been altered either through breeding, not having bacterial biodiversity or biomass compared to the ones on the right. So when you have a whole lot of biology in there, they help to initiate these root hairs, which is all going to increase your water and nutrient uptake. So if you don't have that, then you're going to have a reduction in your disease protection, and you're going to have a reduction in nutrient and water uptake, right? So absolutely critical. So this image here on the right-hand side, so on the left-hand side is wheat, and on the right-hand side is corazon wheat, also known as kamut, right? So it's an ancient grain. It hasn't been modified the same way that the wheat has on the left, but see the difference in its rhizosheath, see the difference in aggregation, massive rooting systems. And what we've done is we've done this to these wheat plants on the left. Sorry, puppy's got a bone. I'll just take it off here. We've modified these. We've messed with plants by focusing just purely on production instead of how do we have systems that have our biological companions, are setting soils up, are building carbon, and are building water holding capacity, right? You can grow corazon wheat in Australia. I get really interested in it. It's a grain that cooks like flour, but doesn't have the gluten in it. So celiac people can eat it, and it tastes just like flour. I mean, just like wheat. I love it. She's doing that on purpose, I'm sure. All right, I'm just aware of time. We've got half an hour. I'll touch on this. Okay, I won't, no. I just want to talk a little bit, just to go from the epigenetics into what some of the herbicides, pesticides, and fungicides are doing. So neonicotinoids, for instance, just that one seed treatment that's used to control insects alters 600 genes in the plant. It is an epigenetic disruptor. These genes, some of them are responsible for plant defense and cell wall structure. They also reduce secondary metabolites, so the plant defense molecules like phenolic, salicylic, jasmonic acids, or hormones rather. It also binds to the minerals that the plants need for defense. It's just fascinating to me that we're using these controls that create secondary problems. So it's like, use my product, you're going to need these other products. And now we're on this treadmill, because if you don't use them, you're going to have more fungal diseases, even though you put on an insecticide, which is fascinating. What else do I want to say about that? Yeah, so for every one benefit from the use of neonicotinoids, there's 100 costs. So it's one of the lowest cost to benefit ratios of all the inputs that we use is the neonicotinoids. So if you can do anything for your system, take those seed treatments out. They also reduce mycorrhizae, so that's AMF, and PGP stands for plant growth promoting organisms, so in the soil. And they suppress beneficial insects' natural immunity. They reduce plant bricks, reduce sap pH, and increase free amino acids, which are what ring the dinner bell for other types of insects. So these insecticides, fungicides, and pesticides disrupt how that plant can function naturally and set it up for more weeds, pests, and diseases. It's fascinating. Do this for yourself. If you have a sap pH meter, is if you apply a fungicide, go out and actually test those plants after you have applied the fungicide, and what you'll see is that pH will drop. We were working in a vineyard, and the pH was dropping down to 3.6. I mean, it's basically, it's not quite battery acid, but it ain't good for the plants. And that's what would happen after we apply a fungicide. So it's very interesting. So insects and disease pressures directly and indirectly relate to soil health and nutrition. So they avoid, so insects don't just eat everything. They avoid complete proteins and high bricks food. And if they are forced to eat it, this study showed that it reduced their ability to breed, they laid less eggs, and then their juveniles were actually stunted. They grew smaller if you force them to feed it, all right? They will choose not to feed on complete protein, high bricks food. And so this graph, which was from 30 different properties in New Zealand, showed like, so it's an insect pressure index that as insect pressure went through, it was extreme at low bricks. So yeah, low bricks, high insect pressure. And at high bricks, we're seeing very, very low insect pressure. And there's actually a very direct correlation. I mean, often you see these graphs and they don't correlate at all, and you're like, how do they say there's a correlation? There's a direct correlation that as we lift plant bricks and plant function, insect pressure goes down. So if you're dealing with red spider mites, any kind of root chewing insects, nematodes, there's a direct relationship with bricks. So if you want to increase your plant defense, we're going to provide, we're going to help complete those proteins. So if there's a breakdown in that synthesis of proteins in the actual plants, they will form what we call the funny proteins or incomplete proteins. So we can use biological stimulants, humic or fulvic acid or milk will actually help to complete those proteins. Molybdenum and boron play a role in this in complexing these proteins. So take a look, do plant tissue tests, have a look. Lift your bricks and build soil health. Okay, I think I was going to stop this presentation and just go to a Q&A because there's been a lot of questions, and I think that would be better than me just talking at you.
Thanks, Nicole. So do you want me to ask some of the questions that were in the chat box?
Yeah.
So one of them was, I sort of think you answered this a little bit, but why isn't the term humus never used anymore?
I don't think it's not that it's never used anymore. It's just that there's a scientific debate going on about actually there's no such thing as humus. All that humus is the, you know, it's just organic matter. It's just really, really super fine. So the scientists haven't actually agreed. And if you look at the amount of papers that are coming out still using the word humus, I don't think the word's lost. I like the word humus. I think gardeners love it. We know what we mean. It's the lovely, sticky, dark brown carbon material in your soil. So I think we just hijack those scientists and we keep using it. But it's very difficult. Like, you can't define humus based on any kind of chemistry because it's going to depend on what died. This is an Australian shepherd. She's being very Australian right now, being troublesome. Yeah, so let's keep using it. I'm not worried.
Yep. So someone's also asked, where can we source more ancient plant types instead of the new cultivars? Is that just based on regions, or is there any recommendations you have there?
Potentially based on regions, or finding some of the old school farmers, or even some of the organic guys that have managed to keep some of the older seed, or guys that are keeping their own seed. Yeah, and getting curious about some of that stuff. Where can we find it? It's still around. We haven't totally lost. Yeah, totally lost the old seeds.
One of the questions sent in prior to the event was, could you explain what CEC means?
Oh yeah, So that's cation exchange capacity, which CEC is basically a measurement of how big your bank account is. So, you know, have you got a little sandy soil, which is a piggy bank, or have you got a great big black clay soil, which is your bank vault, right? So CEC just basically gives you an idea of how much moisture and water can I hold on to, and it's all about surface area. So if you take a teaspoon of sand and you had a teeny tiny measuring tape and you measure the outside of that sand on the teaspoon, it's about a square meter of soils of surface, all right? So that tells you how much water and nutrient. If you do the same thing with a teaspoon of clay, stretch it out with your little measuring tape, it's about a football field in size. So how much water and nutrient. Now, if you did the same thing with these humic compounds, stretched it all out, it's about 4 football fields. So you can see why there's such a drive for organic materials and humus, is that we want to build that sticky ability to hold on to water and nutrients. You going to behave?
I think you said at one point may have mentioned using milk on, why use milk on the soil? Isn't it a food that favours bacteria?
Yeah, but it feeds a lot of lactobacillus. So we find that it does help to complex those free nitrogens in the plant. So I like it. I think it, but potentially you could add a bit of humic acid or a fish hydrolysate if you were spraying, just to balance that out a bit. But yeah, we see good results with it. And it's got sugar, it's got phosphorus, it's got calcium. Yeah.
Yeah. Someone's also asked, so can you influence, this is for the orchardist people out there, can you influence fruit set with soil biology? So this is in regard to someone who may have had a light crop of plums and persimmons this year. Yeah.
Yep, you can. You got to look at what's happening, you know, when we're dealing with trees. Obviously we're dealing with the season before, you know, what were the conditions, you know, and that bienniality that we see in a lot of crops now, like these massive crops and then nothing, is really plant stress. So those plants really are putting a lot of energy into producing those big crops, and then we have a deficit the following season. So it's how do we help support that plant? And what you find is most orchards, I think nearly all of them, unless they're working with us, or, you know, working with some kind of regenerative program, are very, very bacterial dominated. It is not the condition for those plants. So yeah, certainly fruit set's going to have a role with, you know, your trace elements and mineral mobilization. So until your biology are functioning, then you might need to do foliars to support that.
Yeah. So Greg on the chat box has asked, they have a lot of barley grass in a long-term hay paddock, which they've taken over. Yeah. What management tips would you suggest for this barley grass?
So barley grass is quite interesting in that it can be 2 things. It can either be a nitrate indicator or it can be a sodic indicator. So it's telling us that there's a rising salinity perhaps in those fields. So you've got to get really interested. So they will, it actually has a mycorrhizae that can deal with specifically these sodic soils and will recolonize sodic soils with mycorrhizae, which is all very helpful, but not very helpful when you're trying to grow a crop. So you want to, first of all, get interested in, is it nitrates or is it sodic? And if it's, oh, vicious. I have to put the dog outside. It's getting a bit hypo. Yeah, you want to figure out, you know, is it nitrates or is it sodium? And then start addressing that at the root cause. Like, okay, so, and that's where those humics and those vermicasts can be so helpful, is we can do seed dressings, which is what the rest of that PowerPoint was. I'm sorry, I just got a bit ambitious. Is, you know, we can affect that epigenetics. We can affect what's happening with sodic soils by doing seed dressings to start to shift that away from whatever's causing that condition. So again, bacterial dominated soils, and it's one or other of those conditions, and then you've got to start to get to work. But if you've got sodic soils and you're doing seed dressings and we're starting to get that rhizosheath development, those plants can actually now function in what were more sodic soils, and we're seeing those sodic areas shrinking on operations that are using these practices. And the Haggertys are probably good people. I mean, everyone's busy at the moment, but for people to talk to about what they're doing with their sodic areas and why couching.
So there was actually a question put forward about the Haggerty. So in your book, you've mentioned that they've put wormweed out with their herbicide. What measurements would you recommend of wormweed to herbicide? And they're putting it on, they've said here, they're putting it on giant rat's tail.
Yeah. Yeah. So yeah, I mean, it's probably a good idea for the giant rat's tail, which is another one of our bacterial indicators. Yeah, it depends. Like if you're talking about glyphosate, then I generally go off like a ratio, like 1 part glyphosate, 1 part vermicast to 4 parts glyphosate. So that ratio we're getting good results with, or just aim for a litre per hectare of the worm tea in your herbicides. But yeah, it increases cell wall permeability and it increases your kill. So, I mean, and then you're feeding beneficial microbiology. So I think it's a bit of a no-brainer really if you're herbiciding to put in a worm extract or a fulvic acid. So there's fulvic and humic acids in worm extracts. That's why they get that color.
Yeah. Can compost extract be used as fungi food instead of fish when applying calcium hydroxide as a foliar application?
Can those compost teas or extracts? Tea?
Extract.
Yeah, so if it's a high fungal extract, then yes. So you're supplying those quorum signals to turn on the fungi. So potentially yes, if you know that you have, like some of you are making Johnson Sue compost, Google that if you're not sure what that is. But those composts are very fungal dominant, and then that's going to help to provide the stimulant or the catalyst or the signal for fungi. Yeah, when you're putting it on with a calcium hydroxide, so what you're saying, you want to make sure it's very, very dilute and the hydroxide is nicely mixed before you add in the extract to that. And then potentially it's going to kill a lot of the biology, but it's not going to kill the proteins and the signaling molecules or the spores. So yeah, you could do that.
Okay. What is your opinion regarding the WA trend to dry seed? And is there consequences to the soil structure and its health?
I'm not totally clear on what other practices are happening when they're dry seeding. Like, are they putting something else down? I know you're putting it into dry soil, but I don't understand why that has a negative consequence for soil health. Why are they thinking it does?
I'm not 100% sure.
Answer that in the chat.
Yeah.
Okay, next one. Oh, the speed of operation? Not sure.
We'll probably skip that one. If someone wants to put in the chat explaining that a little bit more, that would be helpful.
Yeah.
But so someone's also said they're next door to conventionally run dairy and have an issue with nettles encroaching.
Yeah. So nettles, if you remember from that list, they are your nitrate indicators. So it's not the neighbors, it's whatever is coming onto your property. So if it looks like it's running in, then there's probably nitrates coming off the neighbor's place. And especially here, conventionally run dairy, now that I think dairy, yes, they've got nitrates. So you could just, again, milk and humates will help to mop that up. Or you could just let the nettles grow. I mean, they're something you could dry and harvest, and compost. They're very high in silica, and you know, think about a lot of Western Australia or Australians are very low in silica. So you know, potentially it's something that yeah, you could put in through your compost piles. And if your kids are being really naughty, you can harvest it.
Someone's looking at purchasing a property. What would they be looking at in regards to soil health, like when you're going out to purchase a place?
Dig some holes. I find it's your biggest investment and people don't dig holes. But yeah, certainly look for what your soil carbon levels are, and then just get interested if there is any kind of toxicity. If you've had a very long history of chemical use, we can see some issues arising, but mainly we're seeing those issues because people aren't putting fulvics or worm extracts out. We find if you can take on really degraded land and still rehabilitate it. So yeah, you know, if there's been sheep dip, old house sites, high lead, something like that. I'd be kind of interested in seeing things like that. But dig holes, have a look, see, have you got some massive hard pans? But we're finding that pretty much every challenge that we're dealing with, we're able to overcome. It's just, it's been degraded for a very long time and you've blown all your topsoil away, then it's going to be a slower journey than a place that has been looked after and we have aggregate structure or we have deep rooting depths with good rhizosheaths. So, you know, dig holes, dig holes.
I think that's some of the, like with the dry seeding, people are nervous that it's the erosion or like the dust that is related, like the dust storms and stuff that might come out of it. So I suppose that's, that's why you want to be feeding your fungi in the previous season and then putting seed treatments on with those seeds when you're seeding them. But you're still going to get dust unless you've been doing a biological program for a little bit. But if you think, we're talking about that surface area with your CEC, the finest stuff, that humus is the first stuff to blow away. So when you see someone going over land, stuff all blowing, that's your humus. Like they're losing the most valuable component. It's the first stuff to blow away. So yeah. So I mean, potentially if you could put something down with those seed dressings, and a dry seed dressing, I guess, if you're seeding dry, and just set that community up to take off when you hit the ground. Yeah, I was going to show some pictures of us dry seeding in Montana. We're putting dried vermicast and seaweed into the fertilizer bin and putting multi-species perennials or annuals, depending on what we're doing, down into existing rangeland and getting some really great results. I'm just waiting on some more this season, but the last few seasons we've done some interesting work with dried vermicast to just set that plant up. Yeah. What would be your must-do tip for someone dipping their toe in the water for the first time regarding regen ag? I think they're in a broadacre cropping enterprise.
Read my book. Or listen to the audio book, actually, so it is on audio. The must-do tip is find someone that you can work alongside. Like, don't try and fumble and bumble your way through this without just a little bit of support while you get going, especially with broadacre cropping. It's not that these systems are risk-free. I mean, I certainly think it's less risky than the current conventional system, but I think finding a mentor, finding a group that's been doing this for a while. I really like Vic No-Till and what they're doing in terms of supporting people. So find yourself a bit of a support network that's experienced so that you feel confident and you can take some of these steps. But certainly starting to, just build your observation skills, start to ask those questions. Why, why do I have these diseases? Why have I got weeds? And just get really curious about what's happening in your system, and don't just take here's the Band-Aid, we're going to put this on top. Like, this is not going to cut it anymore, and we need to be making money out there, or we're just not going to be able to continue to do this. Yeah.
I suppose that's where, like, RegenWA here in Western Australia really want to try and connect like-minded people and put out case studies and try and have webinars like this. So hoping we're going to get some WA farmers on board and they can share their story as well, so then people are getting some understanding about what they might be able to go out and do on their properties.
Totally.
There was a question also about your thoughts on utilising natural capital accounting to help understand land condition and asset value.
Yeah, so I think you're talking more about ecosystem services values. So we are seeing more of that coming about. So there is a massive global ecosystem services accounting network that's developing. How do you value this, and then who's gonna pay for that as well? But they did a study in New Zealand comparing ecosystem services between regenerative dairy and conventional dairy. The conventional dairy was providing about $62 a hectare in ecosystem services. So that's, water quality, greenhouse gases, food quality, pollination, diversity, all of that. The regenerative operation was providing $4,500 per hectare of ecosystem services. So yeah, water quality was cleaner, reduction in greenhouse gases, diversity, everything. So from $62 to $4,500, we have the potential in Regen Ag to significantly increase those ecosystem services. And now that there's, I guess, philanthropic people, depends what you believe in around market forces, if you want to be involved in something like that. But you get those services because, you know, you've got more water holding capacity. So yeah, there's a whole debate about markets. Yeah.
It's a bit broad topic.
Yeah, it's a very big topic.
Fish hydros.
Oh, I answered that one. I did.
Oh, okay, sorry, my bad. What's happening with onion weed and Guildford grass? We already know that soil is compacted, so it is an indication of this also an indication of mineral deficiency. At certain times of the year, the bulb is very sweet. Could it be a sugar store to increase bacterial activity?
I don't know about that. That's really interesting. Potentially could be. Definitely bacterial dominated soils. The mineral deficiencies, we did some work with another type of onion grass, but we shifted it. Like the bulbs were still there, but it wasn't growing above ground. And we did that through the application of a fungal based compost. It was really cool. But yeah, it's moving that. And then I would be taking, so when the grasses are growing in your Guildford grass, take a sample, send it into the lab and have a look at what is it mining? What is it high in? Is it high in boron? Is it high in zinc? And then actually applying zinc or boron back to the soil, because it's thriving because that's low in the soil, and it's what we call a dynamic accumulator. It's helping to bring those nutrients up and make them available. And you could wait 100 years and it's going to fix your soil, but I'm not that patient. So yeah, do a tissue test of any weed that you're interested in and curious about what's it trying to tell me, and then do a tissue test, a plant leaf test of a grass that you prefer or you want to see more of, or, you know, if you're growing trees or whatever you're growing, is send in those samples and look at where are they significantly different and where is that weed significantly higher, whatever a weed is. Yeah.
Do you prefer vermicompost over thermal compost? Is it easier to get a high quality product from worms?
You can stuff both of them up quite badly. Most people make a very bacterial dominated vermicast. So the trick really is making sure you're feeding a lot of carbon materials, and people 99% of the time are not. And if you look at the Johnson Sioux and how fungal that is and how fantastic that product is, it's actually got worms at the end process. And I was talking to David Johnson, and he was in agreement that actually it's a fungal based vermicast. It's just that you go through that composting process and then put the worms in there. So I don't know if it's easier. I think it's easier because I don't have any trouble doing it, but yeah, feed them lots of woody type materials. There's a question in here about red gums. I think some of the gum species that are going to compost better and be better in the compost, have a look at, because some gums have total exclusion zones, like they don't have any grass growing underneath them, and some species don't, and you'll know what their names are. I don't. But if you've got grass that comes right up to the trunk, those types of gum are going to be more beneficial in a compost than the ones that don't. And the ones that don't, you're just going to have to pre-compost it, allow it to break down, spray some fish on it just to get it breaking down before you feed it to worms, or yeah, you put it in your compost.
So soil health, is it like when you're looking at your soil, do you think of it differently when you're working in a flood irrigation system?
Yeah, flood irrigation systems are tricky because it's, you know, it's just really tricky to make sure that you're not creating anaerobic zones and you're not overwatering, and the tendency really is to overwater. But I'm seeing people actually putting biological, like just pouring gallons of fish into the gates, and actually when you first flood, actually pushing fish out to, you know, so you're not having to go out and spray it. So there are some opportunities, I guess, to flood irrigation. But yeah, being really, really on top of water management is key. Yeah, it's probably one of the ones that we see more issues around waterlogging. So getting those types of stimulants to open it up, or having plant species that are going to help open up the soil. Yeah.
Yeah. What about couch grass in a vineyard, especially if it's an organic one?
People always want to talk about their weeds. Yeah, so couch grass, any of those primitive grasses, again, bacterial soils, but couch is a big one around calcium and calcium mobility. So feeding fungi, applying some kind of, so an organic orchard, we could do those white washes, and particularly in orchards, I'd probably be doing that as a hygiene cleanup and to set the orchard up for the following season. So I'm not sure if you're growing in winter or summer, depends where you are, but so when you harvest that last apple or whatever you're growing, instead of going on holiday, which I find most people do, go and put a hygiene spray out with a bit of calcium in it, spray it on the actual branches, get it on the ground, put some fish hydrolysate in with that, bit of humic, to help stimulate the fungi and stimulate and lift calcium, and then and just get your eyes off the weeds, guys. So focus on what it is that you do want, what are the conditions that that plant needs, and start to push it that way. So most people's orchards are far too bacterial. So like kikuya, same thing, very, very bacterial dominated. And as we start to shift it towards more fungal, we end up with more advanced, softer grass species, and you'll see that noticeable change. And in orchards, I want to see companion species for those trees. So we did some interesting work in the Hawke's Bay in New Zealand, and what we saw was as that soil shifted towards more fungal, what started to grow were wild strawberries and alyssum and clovers and low-growing species that would naturally be companions to those types of trees instead of primitive grasses. You're farming for the wrong species. So how do we push it biologically towards the species that we want? Yeah.
There's one more question here that I really wanted to ask. We've been making a bioreactor, and after 12 months, we are wanting to know the best way to apply it to get the best results.
Mm-hmm. So that question probably comes down to how much land area do you have. So if you're wanting to treat as much area as possible, you're gonna put it through an extractor, or I like slurry sprayers, but slurry, you're probably going to put 20 kilos out, whereas if you're doing an extract, it might be like 2 kilos, and you're going to cover as much ground as you possibly can. If you're putting it out as a solid, then you're going to be putting out potentially like 700 kilos a hectare. So you can see why the slurries and the extracts, you're going to be able to go further. But if you're putting it out as a solid, you're not going to have to repeat that solid application for at least 7 years. And that work has been done in vermiculture and composting to show if you get a really good fungal dominant diverse compost, it's a one-off application for 7 years. So depends on your scale. If you're large scale, yep, you're going to get a slurry or an extractor, and yeah, you can make them quite simply.
So, well, there's heaps, still lots of questions, I suppose.
And we got 1 minute.
1 minute.
What? Oh, I'm really, you guys are still with us. Thank you.
So one question here, it says, what regen ag strategies could we use when market gardening, where we have permanent beds with successive plantings of annual plants?
Yeah, so some of the large scale market gardens I'm working with, we have rolling cover crops, fallows, where you allow that ground to recover, or you have a rebuilding phase. Seed treatments, you know, wood chips go really well in some of these gardens. So, you know, like just trying to push it, because everything that we do really is a disturbance, and we push it more and more bacterial, especially in vegetable production where you might be cultivating or you might be spraying. Thanks, Ewan. Then you got to look at, okay, everything I'm doing, I'm pushing it backwards. So everything that I want to do is to counteract that. So I'm going to put something down when I'm seeding, or I'm going to put something down when I'm harvesting, or if I'm cultivating, I'm going to keep pushing it back towards more fungal. Some of the guys I work with have permanent alley crops of, say, like lavender or rosemary or something that's pushing mycorrhizae out into that soil system and just helping to keep, you know, a bit of perenniality in systems that are highly modified. So yeah, just think, how do I feed my fungi?
I think we will wrap it up there. I think for anyone who hasn't yet got a hold of her book or her audio book, it's really good, especially for answering a lot of those things. And like you said at the beginning, there is a story in it, but it's got so much technical and case studies in it and examples. I think it's a really good advice. And some of the comments that people have put up saying they've read it many times and there's tabs in many of the pages, and they all think it's a great read for those who have read it. So a good resource. Thank you so much, Nicole, for spending your time with us this morning and in your evening. I think it's been really valuable. We at RegenWA, we are trying to make this session sort of available as a resource, so I'll be able to send that link out to everyone as well as the last one, and we'll continue running webinars like this. So yeah, thank you so much. Yeah, enjoy the rest of your night, and your little puppy was the delightful.
Not helpful at all. All right, thanks guys, thanks for being on the chat, and yeah, thank you for grabbing a copy of the book. Have a great day and a great season.
Thanks, Nicole. Talk to you soon.
Thanks, guys. See ya.